It seems like the never-ending lecture, doesn't it? Do you remember that movie, The Never-Ending Story? Or again, am I showing my age to you? I hope that you've seen The Never-Ending Story. It was one of my favorites growing up. But this is the never-ending story of DNA. At least that's what it sounds like. How much more can we talk about this horrible DNA structure and how it's made and what it does? All right, so on the screen right now, you are seeing a three-dimensional computerized structure of DNA. And this is kind of what the thing looks like, right? So if I just blow this up, there is some movement to this molecule. And as we can see around the outside, you see a lot of those white and reddish and orangey yellow looking things that go up and around and around and around. That is your sugar backbone. And there's two strands. One's kind of coming toward me and one's going away from me. And as this thing turns, I can see those two strands interact with each other. Okay, so that's the sugar backbone. And then on the center, I see the steps of the ladder, right? So as I go up, I'm seeing these perfectly flat areas. And that shape never really changes. That's the presence of the bases. The A, the T's, the G's, and the C's. All the way up they go. As long as 6 foot in length. If that's how big this molecule of DNA is, all the way up, and it will twist and twist and twist around, very similar to a slinky. And we kind of see that. We see the minor groove, and we also see the major groove. So if you take a look, we see a really big spot that's here, and then as this molecule twists, well, we get a smaller spot that happens, and then a bigger spot that happens, and then a bigger, smaller, bigger, smaller. And that is what we refer to as the major and the minor groove. So this is the stuff that we're working with. When I'm in a forensics laboratory, and I'm taking a DNA sample of someone, it's this right here that I'm working with. This molecule could be so big that I could visually see it with my own eyes. That's pretty impressive. And it's up to me to analyze this thing, because this strand, this molecule right here, from someone's mouth, or from someone's blood sample, or maybe even from someone's hair sample, that molecule is going to be different from person to person. And it's all about the bases that are on the inside of this thing. What bases are present and what order are they present in? That is going to determine the differences between our DNA strands from each other. All right, so there's a three-dimensional structure of DNA right there for you. All right. So, here's just another version of that. It's not fancy. It's not moving around. But again, you see the sugar backbone that goes around and around in the helix. So we're seeing it come around, and then it goes up, and then it twists, and it goes around again. All over and over and over. We also see the base pairs once more in the center of these. There they are. That's what makes the runs of the ladder. And they're pretty symmetrical again all the way up and down. This diagram is also showing you the hydrogen bonds, just so we don't forget it. So between the A and the T, we have 2. And between the G and the C, we have 3. Our pyrimidines and our purines. Here's another depiction of a DNA molecule. This is what we would call our space-filling model, because each one of these atoms, well, it's not just a carbon C that's connected by a line, all right? That line is there just to denote a bond has formed. These things are actually physical. They're kind of lobe. They're kind of spherical in a way. At least that's how we depict them. And this is the molecule as it's filled. How much space is it taking up? So what you're seeing on this diagram is the green strand that kind of comes up, and then this is actually going to twist around the back end, and it's going to come back up again. That is one of our sugar backbones, the ribose or deoxyribose and the phosphates. The yellow strand is the complementary version. So this is another strand of sugar and phosphate that comes around, and then it will twist upon itself just like the other one does. And that other strand runs anti-parallel, right? So we said if the green one goes to 5' up here at the top and the bottom one is 3', then the yellow one runs in the opposite direction. This would be the 3' end, and as it goes down, this would be the 5' end. Anti-parallel. Finally, in the middle, you're seeing all of these gray and red and blue orbs just kind of crammed and smacked right dab in the middle, all the way up as we go. Those are our base pairs, our A, T's, G's, and C's. Notice how the sugar phosphate backbone somewhat protects this strand of bases that are in the middle. It's kind of like a bodyguard for them, right? And they are a bodyguard. Remember, we said that these are highly negative right here, and they are going to push away nucleophiles if they come into the picture. We don't want this DNA to react. We want this to be very stable. And that is one of the ways that it gets its stability, is this highly negative area on the outside. It pushes all of them away, every one of them. So DNA is going to be pretty important. All the genetic information that you have, that you carry, will be involved in the DNA. It's not sugar now. We know that. So 3 functions have to happen here. Number 1, we have to make sure that DNA stays protected. And we kind of see how it's done that on its own. It is taking care of itself. Long before we came around, DNA was there. DNA took care of itself. It stabilized itself. It knew how to protect itself without you getting involved. In a way, it's kind of smarter than we are. Number 2, it has to stay preserved. So once it is made, how on earth do we keep it? We don't want it to change that much, and it won't. The DNA that you have as a baby will be the same DNA that you have on the day that you pass away. And that DNA is not going to change. And number 3, it has to survive. It has to be passed on. So in order for this process to work, DNA had to be clever enough to figure out how to keep this going. If we have genetic information, how can we, along the years, in the future, our children's children's children, how can we preserve as much as possible? We don't really want this to go away, meaning that in the beginning, these people might have had DNA, but over a course of years, we did not need that DNA to disappear and go away. It had a purpose. How can it continue that purpose on and on and on and on? So in order for this to happen, DNA had to learn how to copy itself. And in order to copy, there's a message that needs to be transferred. So how do we, what we call, decode this message? And how does the message make sense? So there's 3 things that has to go on with DNA. Number 1, it has to replicate. How does it replicate? Well, DNA has information. It's like an ancestor. And that information is passed on to the RNA molecule. So think about sitting around at the feet of your grandparents when you were younger maybe, and they would tell you all of these stories about what happened to them in the past. All right, well, your grandparents are the DNA. You're those kids that are sitting around the rocking chair at their feet, and they are telling you what has happened. So that way you have a memory of it. You have an idea of what's going on. You've had an idea of what came before you. And that's the purpose and the role of DNA and RNA. Now, once you're told that story, it's up to you to transcribe it. Meaning, you read it and you decode it. You make sense of that story. So if your grandmother wrote down a recipe for you before she passed, and she gave you that recipe and said, carry this recipe on through the family lineage, well, you would read that recipe and you would try to understand that recipe. And that's what RNA does. It says, okay, this is the remnant. This is what I have. This is the memory. And this memory, I need to read it. I need to understand it. So that way, in step number 3, I can translate it. I can tell other people, maybe my own children, how to replicate that recipe that Granny used to do way back in the day. There's your 3 functions, and they're closely tied together. It needs to replicate, it needs to transcribe, and it needs to translate. And that's the role DNA plays, and that's also the role that RNA plays. The reason that I'm mentioning this is because in a forensics laboratory, copy is a very good word for us to use. Because chances are we will find a DNA sample, and we might not have enough DNA in order to do things with. And if that's the case, we need to copy that story. We need to take that DNA sample and we need to copy it on our own. So that way we will have enough to do something with. So this copy idea is very important to us. Now, the RNA stuff, that can come a little bit later. That's more biology type. I'm going to give you a little rundown of how this process works, but that role never goes away. What we want to focus on, though, is the DNA sample. So let's talk about the first one, this replication. We now know all genetic information is contained within DNA. The human DNA, how many base pairs are we talking about? 3.1 billion. That's right. 3.1 billion base pairs. Remember, I said this DNA strand can go up to 6 foot in length. If this molecule is so tiny that you cannot see it with the naked eyeball, but by the time all of these base pairs come into the picture and elongate that strand, and that strand gets to 6 feet, you can imagine the number of base pairs that's contained here. And 3.1 billion. That's even hard to believe. Look at the number of placeholders here. Imagine $3.1 billion that you have in your bank account. I don't think anyone could imagine the possibility of what they could do with that money. No longer millions, billions with a B. So there's got to be a way that we can look at DNA and we can talk about it in segments. We can't talk about 3.1 billion base pairs at one time. We've got to be able to go in and talk about smaller pieces of it at a time. And now we are getting a little bit bigger. Remember, I told you, chemists have a different perspective than biologists do. We are slowly working ourselves up from the foundation level. We talked about base pairs, then we talked about sugars, then we talked about the sugars connecting, then we talked about the base pairs going side by side, and then we talked about the formation of the DNA molecule, and now we know 3.1 billion base pairs. We now are going to go another step up. The way that I'll talk about segments of DNA, this is called a gene. And normally they're about several thousand. You see that in parentheses right here. About several thousand base pairs. And that segment of the DNA molecule is going to be called a gene. Well, all of those segments can come together. We can talk about multiple segments because remember, 3.1 billion base pairs, that's quite a bit. And those genes can come together and make what we call a chromosome. And each chromosome has a pair. 23 pairs. 46 total chromosomes. All right, so if you're in a biology class, this chromosome is typically kind of this X thing that you typically draw. Maybe something that looks like this and something that looks like this. That's another representation of it. So we are getting a bigger and bigger picture now. We're starting smaller at the DNA molecule. We now know that there's billions of base pairs that are lined up from end to end. We now know that it's almost impossible for us to go through and talk about all of them or even realize that all of their purposes. So we're going to cut that DNA segment down into segments. These segments are called genes, and the genes are going to come together and make up what we call a chromosome. And the chromosome has a pair. There's 23 pairs, giving me 46 in total. So more about the replication. So when we talked about the DNA strand, we said there's two strands, and they have these base pairs in the center, ATGCs. And that means that your DNA, if it's caught in a position, if something rips this DNA in half, something disturbed it, DNA has a backup plan. If this gets ripped in half, we are left with one strand with base pairs. But here's the thing. DNA can carbon copy itself. That means if I do something and I rip the strands apart from each other, DNA has survived many, many years. DNA knows how to fix that problem. It now sees this one strand that is left, and it knows the base pairs that are on these ends. A, T, T, G, G. And if it's given base pairs to choose from, this one DNA strand, maybe 5' to 3' here, can actually start assembling the strand that was ripped away on its own. It knows that a C would have went here. It knows a C would have went there. It knows an A would have went there. It knows an A would have went there. It knows a T would have went there. And it can fix itself. This is pretty important because if we're saying that your hereditary information is contained within this molecule, something could destroy this molecule. Something could rip this molecule apart, and it's got to have a backup plan, and that backup plan is copying itself. That carbon copy of the complementary strand. That's all that it needs. It knows what base pairs are there. It knows what base pairs are going to come together to make it happy again, and it will start assembling these things back together again. No big deal for it. We can also play off of this process. These templates, I'm going to put a big star here. These templates are what we will use when we talk about PCR, polymerase chain reaction. We're going to play off of this concept of this DNA self-healing itself. The sequences are there. It knows what sequence would have been there if it does get ripped apart. and it will go back and it will fix those sequences when it can. Now we have some terminology here. If I have two strands that are together, and this is the way it was, no big deal. But I just said that we can go through and we can create a new strand, right? So what if something comes in and erases and rips those two things apart? Well, I told you DNA can fix itself. So it will begin to assemble a new strand slowly and slowly to stabilize itself. And that purple one right there that you're seeing, that's a new strand. If it's the new strand, not quite the original that was there, we're going to call this the daughter strand. It's still double-stranded. It's still there. And this process of it using one half of the DNA molecule to copy the other half, this is going to be called a semi-conservative replication. Semi-conservative. Well, we know what replication means. Well, conservative means kind of save back, right? It's kind of halfway saving back, which means half of the strand is the original, and it's going to use that half of the strand in order to make a copy, a new complementary strand that goes along with it. Again, this is the process of copying DNA molecules in a forensic laboratory. This is what we're playing off of. We've just got to figure out how this process works. If we can figure out what needs to be there in order for this to happen, then guess what I can do as a forensic chemist? I can go in, I can take someone's DNA strand, I can rip it apart, I can allow one half of the DNA strand to replicate and copy. There we go. And then I can take this other half that we ripped apart, and that one will copy all the way down. And now I've taken one strand, and look, I've made two. And then guess what? I could take this strand, separate it again, copy each one of those. And now I have two on that side. And then with this other piece, I can separate those. And they can copy. And now I have four. I started with one. I ripped it apart. I got two. I took two. I ripped those apart. And then I got a total of four. Then I'm going to take those four. And I can rip those apart. Replicate each one of those. And then I can end up with 8. Then I can take 8. Rip all of those apart. Let them copy. Now you can see exponentially how we can replicate and copy DNA just from one strand from one person. Again, though, I've got to figure out how this process happens in a laboratory, and that's kind of why we're taking this class. But in order to get there, we've got to understand all of the basics that come along with it. So in the next video, I'm going to stop here. In the next video, we're going to come back. We're going to continue to talk about replication and what we need in order for this to happen.